And as was mentioned, I think there's a poster still out in the hallway on the ARAMP program, so you can take a look at that as a break. But before the break, we have one more talk, not fish habitat, but human habitat. Drinking water is going to be the next talk with Kurt Carpenter with USGS. He's going to be talking about post-fire hyperspectral surveys for periphyton to protect drinking water quality in 3 Cascade Range rivers following the September 2020 wildfires. So help me bring up Kurt to the stage.
Great. Well, hi, everybody. Thanks so much for having such a great compilation of talks. And we have been drinking from the fire hose, but it's been really great to get the full 360-degree view here. So I'm going to try to round that out with a talk here today on periphyton, which are benthic algae. There we go. So just before I start, I wanted to acknowledge all the different folks that have been involved in this project. All USGSers from different water science centers across the country that bring different aspects of this project. Different folks involved with remote sensing and flying drones and processing of data and microscopy and other aspects of the project. So periphyton, again, are attached benthic algae that can cause some pretty significant issues in our rivers already, actually. These include filamentous green algae. We've seen some pictures in some of the talks already where you see stringers of green algae. Here's some photographs. Another real common type of periphyton are stalked diatoms. You're probably familiar with the regular flavor of diatoms, but the stalked diatoms are kind of a unique organism related to Didymosphenia that you might have heard about that causes big high biomass blooms in New Zealand and in Idaho and other rivers. They call it rock snot. Well, we have a really similar organism, Cimbella, and also Gomphoneus that are similar stalked diatoms that cause the middle picture on the bottom there. Pretty impacted habitat. You don't find very many invertebrates with these stalked diatoms, and so we're tracking those. They're some of the largest diatoms. You can actually see them with the naked eye if you look really closely. And then also the cyanobacteria, right? So cyanobacteria we can thank for all the oxygen that we breathe, but they also can cause some pretty significant issues with taste and odors and disinfection byproducts and cyanotoxins. This spring or early summer I'll release a report and a data release on an extensive study of cyanotoxins in the Cascades. Suffice to say that we find them pretty much all over the Cascades. So ask me about that maybe later. So these benthic algae can have significant effects on higher trophic levels, like benthic invertebrates by affecting the habitat, food webs, and then the high pH, which is an issue that I've recognized and talked about for years, but we still haven't fully gotten to the bottom of it. So one of the reasons for studying the periphyton beyond the obvious list and impacts to drinking water and food webs was the fire. So I've been tracking periphyton growth with the drinking water providers, eWeb, and the Clackamas River water provider, City of Salem, for a number of decades now. And we see year-to-year variability, of course. The nutrient levels in the Cascades aren't very high typically, but after the fires, we saw very large increases in nitrate levels. And normally when we go out and measure nitrate in streams that have periphyton, we don't find any. It's the levels are below detection oftentimes. And but after the fires, and the top graph here shows the 4 streams where we focused some studies right after the fire. We stood up some continuous stream flow monitors and then also some continuous water quality monitors looking at pH, dissolved oxygen, turbidity, and those sorts of things. And so we have 4 different tributaries, Little North Santiam, Fish Creek in the Clackamas, Gate Creek in the Mackenzie, and then also Rock Creek, which we heard about earlier today. And what you see here is average concentrations coming in at about 0.5 milligram per liter, which is quite high. When you compare, less than maybe 20 or 30 micrograms per liter during the growing season, and now we're seeing, 0.5 milligram with peak concentrations getting up to 1.5. And you see these 4 tributaries. So these are all heavily burned watersheds, 75+% had high severity burn. But what you see is pretty similarly high concentrations in the peaks. And what's interesting about those peaks is that, we have auto samplers out that collect samples over the course of the storm, and, we see the turbidity and the dissolved organic carbon, which we're also looking at, peak. And then as the sort of the storm peak goes away, the nitrate stays. Those highest nitrate concentrations are in the aftermath of the runoff. It's really that recession, the receding limb, and that can go on for quite a period of time. And so as that happens, the water's clearing up, and that's just ideal conditions to grow algae. So you see similar situation with phosphorus, much elevated concentrations of phosphorus, but we have high levels of phosphorus in the Cascades, right? The high Cascades are a natural geologic source of phosphorus, and I think there's some evidence that the land management can transport phosphorus to the rivers, but the source is natural. That erosion can bring some of the phosphorus in, but you add the nitrogen plus the phosphorus, and then also the light environment. We've heard that quite a bit today about how the riparian disappears. And so, the productivity, which tends to be, in the lower parts of these watersheds, think about, Van Note's continuum concept. And as these headwaters have burned and middle reaches of these rivers have burned, now I think the productivity can march up into those tributaries. So we're really interested in tracking periphyton. So as I mentioned, we see large effects on pH. pH is a really important variable. Our cells are all designed to produce a homeostasis, right? We don't like changing conditions, and yet look at what these organisms, fish and other life in the rivers are up against here. We see fluctuations of pH, approaching 2 full pH units each day. So that's quite a challenge, and I would say that's something that has not really been fully fleshed out in terms of the effects on the fish. And these measurements, in this case, taken from the lower Clackamas, are in the middle of the river. So the little fish in the margins where the periphyton is growing are probably experiencing even higher pH. Now, the pH levels get so high partly because of the alkalinity levels are very low, so we don't have a lot of buffering capacity to be able to withstand the fluctuations in pH. And if I didn't mention it, that's because of the photosynthesis by the algae. As they take the carbon dioxide out of the water during the day, the pH goes up. And then when the sun goes down, they continue to respire, and all of the collective stream metabolism brings the pH down. And you can see here exceeding the state standard quite a bit. And this shot here is of some green algae from the Mackenzie River in 2021, so after the fires. So, as I mentioned, a lot of different cyanobacteria. Here are the main ones. We have lots of nostoc in these rivers, a couple different forms. Here's some microcoleus, non-nitrogen fixing mats of cyanobacteria that make, among other things, anatoxin, microcystin, and other toxins. So we're closely tracking this microcoleus, and it's also probably responsible for the many dog deaths that we've seen in recent years. Another common form is oscillatoria, and sometimes we see them, and here's what it looks like under the microscope on the right here. Sometimes we see them on the rocks or on soft sediments, but they can flake up like this. Do I have a laser beam here? This guy here, they can pop up to the top, and then it floats downstream, eddies out, and that's when it can cause some real problems. So one of the challenges of sampling the periphyton is that, especially in a river like the Clackamas or the Mackenzie and the Santiam, is that these rivers are only partially wadable. And so we've been trying to develop some new tools, and here's where the hyperspectral data come in, to be able to sample larger areas, right? Getting a representative sample in an unwadable river can be really challenging. So in this case, we're using satellite data, airplanes. Pretty soon, took a little while to get the FAA permission on the airplane, but I think we've got that now. So hopefully that will be added to the quiver this summer. But then using drones and some other techniques here. So here's Paul Diaz from our Science Center getting ready to fly in Clackamas. And so he's got a hyperspectral camera that, and I'll give you some specs on that in a second here, but that he's flying to be able to collect imagery. He also has just the standard, RGB red, green, blue, high resolution, and then also FLIR camera that we now have capability to put onto this drone there. So the sort of centerpiece for this hyperspectral study is a hyperspectral microscope. So this was put together by USGS colleagues in Reston that partnered with the National Institute of Standards and Technology, or NIST. And so I'm going to talk about a related study first, and then I'll bring it back to the periphyton. So in this case, back in 2022, we published a data release for cyanobacteria that grow in the water column. So these are blooming cyanobacteria that grow in reservoirs and lakes. And this graph over here shows the 12 different genera that we were able to image. And what you can see is that the spectra are similar, but they're different, and they're different enough, which is the key. Here's about half of the 12 that you can see, different colors and different shapes. And so for the hyperspectral data, we're looking at the reflectance, right? It's not like fluorescence, it's actually just what light bounces off of these organisms. And so, we see these different types of signals across a pretty large swath of the spectrum. And, And so the exciting thing here is, when we can tell, let's say, a particular organism here with the hyperspectral microscope, and then if we can collect data from, say, a satellite or an airplane or one of these other platforms, we can compare those and potentially identify the type of algae that grow in a lake or reservoir. And so part of that is building this reference data set. So we've been doing that, and now we're expanding that into periphyton. For the phytoplankton and sort of the more typical classic harmful algal blooms, this slide sort of summarizes the whole process, and we call it SMASH, or SMASH algae. I mean, how great is that, right? It stands for Spectral Mixture Analysis for Surveillance of Harmful Algae, okay, SMASH algae. So what it does is, we've got the spectra here, and then we've been working with the space station camera. Here's this DASIS camera on the space station. And they are like super excited to be working with folks on the ground. They're kind of lonely up there, but, and so Carl Legleiter with the USGS Modeling and Integration Division in Golden put together a workflow process where space station can look at the lake, gets a spectra, it gets compared to our library, and we go, ooh, we think it's species A. And so in this case here, we've got, and there's lots of the lakes that are being monitored by different partners. In this case, USGS has a program with the Bureau of Reclamation where we go out and we collect samples here. And anybody knows Upper Klamath Lake, it's a phanomenon, right? All day long, it's a phenomenon in Upper Klamath Lake. But actually, no. Look at this picture here. This shows many different, each of these colors is a different genus. So it's more complicated than we thought. But the very awesome thing that we discovered was when they went out and they collected a sample from this point, used the space station data to put it all together, all lines point to a phenomenon. And when we discovered this, we were like, wow. I mean, that was like, we were so incredibly excited. We went right to work on the paper because for a long time we've been able to say, yeah, there's a bloom happening in that lake, but now we can say it's a phanomenon bloom, or it's a Dolichospermum bloom, or it's a Microcystis bloom, or maybe it's a harmless green algae bloom. That's going to be a game changer. And so here, this really sort of dials down into kind of the comparison here. So here's what the space station sees when it looked at that pixel, and here's what the microscope sees. So pretty close correlation between those two spectra. So bring it back to the rivers here. So as I mentioned, Carl Legleiter in Colorado, he had conducted a study in the Buffalo River, Arkansas, where they used some worldview satellite data to try to map out the biomass. And so we're wanting to take that one step further by actually collecting a variety of spectra from the periphyton to now see if we can bring this technology to the periphyton. Here's a table that shows the different instruments with the different wavelengths and ranges and number of bands. I mean, it's a really, really rich amount of information, bands that, compared to the 3 bands that we have on our phones. One of the things we'd like to do is simplify this down. Maybe there's a subset of 10 or 15 different metrics or either wavelengths or ratios that we can use to better identify, in this case, periphyton. But this technology, I think, can be used to study invasive macrophytes, health of trees. You know, I mean, if we can see stuff in the water, imagine what you could do on land with these data. And then again, this summer, hopefully, we'll have the Civil Air Patrol on board. So we're conducting what we call these riffle scale hyperspectral surveys. We flying the drones and collecting rocks, collecting spectra above and below the water, scraping the rock, and then getting another image after we scrape the rock to just get that sort of natural substrate spectral signals. And so we've been conducting these different surveys in the Clackamas, in the North Santiam, and in the Mackenzie. And, you know, one of the things that, and I'll digress here just for a second, it doesn't look like I'm running out of time yet, but is that, you know, some years we have a lot of algae and other years we don't. And, you know, I think there's a common misconception that, oh, it's really those low water years where it's really warm. That's really when we have the worst algal problems, but it doesn't really work that way, I don't think. And one of the things I was hoping that we would be able to do from this project with, you know, eventually would be to fly the Civil Air Patrol aircraft and collect data on the distribution of the periphyton in different years with different flows. You know, you can imagine a very low water year, the flows might all be contained within the channel. And then maybe there's just not a lot of habitat and it's kind of moving along pretty fast in the channel. Contrast that with a year where we have a little more water. So the groundwater system's now charged, which I think is a real important source of nitrogen, especially. You know, post-fire may be a little different. We've got a new source out there now, but before, I think it was mostly coming through the ground. And when you have the sort of intermediate rainy, especially the spring seasons, is when we tend to see, you know, maybe more habitat, because now the water's out of the channel and now it's covering the riffles, and that's just like ideal habitat to grow a lot of algae. So the idea over time would be to develop an IFIM, an in-stream flow incremental methodology type of an understanding of the periphyton, so we could better track the trends, because really the trends, I think, are very tightly connected to the flow and precipitation. So just a couple of examples here of the different spectra in the organism. So, if, you know, at the end of the day, if I can tell the difference between green algae versus diatoms versus cyanobacteria, wow, that's a victory. If we can actually use the spectral library to identify the species or the genus level identifications, be so happy for that. What we see are, with the different spectra here, and again, this is for Eulothrix. It's one of the real common green algae. Filamentous green algae that grows in these systems. Here's kind of what the spectra look like, and I've got the chlorophyll A and chlorophyll B absorbents, so not the reflectants, but here it's the absorbents. And, you know, basically we see, you know, low reflectivity here and then here, partly because the chlorophyll A and B are absorbing light in that part of the spectrum, right? Moving to the stalked diatoms. And then we've got field spectra above here, and then the microscope spectra on the bottom. Pretty similar shape, so we're looking at both the magnitude and the shape of those curves in order to make the comparison. So SMASH kind of uses both of those information pieces in order to develop the regression to try to identify what it is, but pretty good agreement there. And then with the cyanobacteria, it's pretty variable. You know, sometimes we see mats like this in the upper left here that are green, but oftentimes they're brown, or less often, you know, kind of greenish blue a little bit. But oftentimes the spectra are flatter. And I don't know if they're just absorbing so much of that light. That's a possibility. You know, very efficient light harvesters. Cyanobacteria can grow deep in a lake. They just don't need as much light. You know, partly they're heterotrophic at times, but they can also just, you know, they've got chlorophyll A, carotenoids, phycocyanin, phycoerythrin, that really gives them a very broad spectra that they can utilize for their growth. So yeah, efficient light harvesters. So the goals of the study are to characterize the different periphyton types using hyperspectral data, expand this reference data set beyond just the planktonic forms into the benthic forms, and we're well on our way to doing that. And then, as I said, use these data and different scales and try to determine, can we just fly the airplane? Do we need to go out and measure with a handheld spectrometer, or can we just do it all with the satellite data? DASIS has some limitations, so that's the International Space Station camera. It has pixel size of about 30 meters, so it's pretty big, So as time goes on and the satellite technology gets better, we should be able to get smaller and smaller and smaller. So maybe we'll be able to do this, from our desktop, from our offices someday, and be able to look at the trends over time. So hopefully I left myself a couple minutes for questions. Yeah. Yes, sir.
Hi, Kurt. Thank you for your work. This is really interesting. I'm wondering if the stuff you learned, say, from the South Santiam can be taken, out of the box and applied to the North Umpqua without additional field work per se, or would it require a lot of boots on the ground field work to apply kind of the spectral stuff you've learned elsewhere and apply it elsewhere? Additionally, like stuff from Upper Klamath to Diamond Lake and a lake system. Yeah, great question. Yeah, thank you for that. So the question was, can we transfer? What's the technology transfer ability here? So Umpqua is a great example. So I've actually done periphyton studies, a number of them in the Umpqua, and we have a lot of the same organisms: Cladophora, Epithemia, a lot of this, nostoc, tons of nostoc in the North Umpqua, and actually. A couple summers ago, we deployed some devices to look at the cyanotoxin, same situation in the North Umpqua that we see in Santiam and the Clackamas and the Mackenzie. So definitely very transferable. Yeah, and same with Detroit, with Diamond Lake. So there's, even though we have a number of different organisms that bloom, it's a pretty short list. There's probably 4 or 5. On that short list, and as I said, we're well on our way to having all those characterized, so we should be able to do that. And actually, I've been working with James Watson at Oregon State here on operationalizing this SMASH. So the idea would be space station or other satellite is collecting data, sending data back, it runs through the SMASH algorithm, and then we get notified, hey, there's a bloom in Blue River Lake, and it looks like it's Stilixspermum, which is a toxin producer. Maybe we need to get out there. So, hopefully with time we'll be using that data and information to guide our research. So very excited, very exciting times we're in right now. Another question? Yeah.
I was curious about the elevated nitrate that you mentioned, and why is it staying elevated longer than the obvious particulate pulse that comes with an event?
Yeah. So question was, why do we see the nitrate elevated the way that we do, and not kind of dissipate after the peak in the turbidity might have gone by? I'm thinking it's probably the groundwater, right? It just takes the flow paths. It's probably coming through the groundwater, and some of these cyanobacteria are nitrogen fixers and some aren't. The microcoleus that I'm more concerned about in terms of the anatoxin and, say, dog exposures don't make their own nitrogen, so they need an external source of nitrogen. And so, all of the nitrate and the phosphate is certainly potential to cause more growth. And actually, a lot of these toxins are pretty nitrogen rich, so that has implications for the amount of toxin that could be produced. So, all right, well, thank you so much.